session, we will discuss the next topic, prokaryotic transcription. In the previous lecture, we discussed what is transcription. It's a process of making RNA from DNA. What is the enzyme required in transcription? DNA dependent RNA polymerase. And from 2 strands of DNA, we make only 1 strand of RNA. Why is it so? That also we discussed. And the boundaries should be defined. So what is that boundary? The unit which undergoes transcription is called as a transcription unit. That also we saw. Now in today's lecture, we will work out with the process of transcription in bacteria, means prokaryotic transcription. We will see it is 6.5.3 types of transcription they gave. We will see prokaryotic transcription in one session, eukaryotic transcription in the next session we will see. Prokaryotic transcription means it is in the bacteria. How does the process of transcription occur in bacteria? Okay, so bacterial cell, if we see, in the bacterial cell, this is the circular double-stranded DNA. This is the circular double-stranded DNA. There are no membrane-bound organelles. There is no nucleus. It is naked chromosome. So that means where is replication occurred in the bacterial cell? In the cytoplasm itself. Then where can transcription occurs? So the DNA is here only. So transcription also occurs in the cytoplasm only. So means from DNA, the RNA molecule will also be made in the cytoplasm only. So if we see the location of transcription in bacteria, it is in the cytoplasm. Since there is no nucleus, it happens in the common compartment cytoplasm. The direction of replication is 5 to 3. The daughter DNA strands are synthesized in 5 to 3. Similarly, the direction of transcription is also 5 to 3. So polarity, direction is called polarity. The polarity of the RNA synthesis goes from 5 prime to 3 prime. Then what is transcription? Synthesis of RNA from DNA. So even bacteria requires all the 3 RNAs. mRNA it requires, rRNA it requires, tRNA it requires. So in bacteria, all the 3 RNAs can be synthesized by one common enzyme. One enzyme will do the synthesis of all the 3 RNAs. So one enzyme will do the synthesis. One enzyme will do the synthesis of all 3 RNAs. What are the 3 RNAs? So one is mRNA, the other one is tRNA, and the third one is rRNA. mRNA copies the message from the DNA. What is mRNA? What it will do? It will copy the message from the dRNA. What will tRNA do? tRNA brings the amino acids. It's an adapter molecule. It carries the amino acids to the sites of protein synthesis. So tRNA, it carries amino acids to the sites of protein synthesis. rRNA is structural because it's making a structure called ribosome. So rRNA functions as a structural molecule, means making ribosomes. rRNA can also be catalytic. That we'll see how it's catalytic in translation mechanism. We'll see. So it is even catalytic. It acts as a catalyst. It acts as a catalyst. So one RNA polymerase enzyme can do the catalysis of all the 3 RNAs like mRNA, rRNA and tRNA. So what is the name of this enzyme? It is DNA Dependent Rna Polymerase. It sees the DNA and makes the RNA. So the enzyme, the complete name of enzyme will be DNA Dependent RNA Polymerase. The enzyme which will catalyze the transcription process is DNA Dependent RNA Polymerase. You can also call it as a transcriptase also. We can also call it as transcriptase. Now this enzyme, DNA Dependent RNA Polymerase, it is made up of many subunits. Hemoglobin is made up of 2 alpha and 2 beta subunits like that. DNA-dependent DNA polymerase is made up of many subunits. So, it is a polymer. What are the subunits? Let's just see. It has 2 alpha subunits, alpha 1, alpha 2. 2 alpha subunits are there. 1 beta subunit and 1 beta prime subunit. 2 alpha, 1 beta prime subunit with 1 omega subunit. So, 2, 3, 4, 5. These 5 subunits are the main things. These 5 together is called as core enzyme. These 5 together is called as core RNA polymerase. Now, these 5 will show transient association, temporary association with either sigma factor, this is rho factor. They show transient association either with the sigma factor or with the rho factor. Now, to the core enzyme, if sigma factor combines, what is this? This is called sigma factor. What is this called? This is called rho factor. Now, the core enzyme transiently, temporarily only, not permanently. The core enzyme make transient association with the sigma factor to start initiation. So, then after that the sigma factor will drop down. Core enzyme only is capable of doing elongation. Then when the terminator sequence comes at the downstream end, so then the rho factor is required. Then again core enzyme transiently binds with the rho factor to do termination. So, the sigma factor, rho factor are coming and they're going out. But core enzyme with these two things is called as core enzyme with sigma factor or with rho factor is called as holoenzyme. Holoenzyme means complete enzyme it is. But the association of RNA polymerase with the factors like sigma factor and rho factor is transient. The association of RNA polymerase, the association of RNA polymerase with sigma factor or rho factor, what did I tell it is? Transient. It is temporary, it is not permanent. Sigma factor is required to identify the promoter. Rho factor is required to identify the terminator. Okay. Now, then steps of transcription if we have to see. What are the steps of transcription? There are 3 steps in transcription. Initiation, elongation, termination. What are the 3 steps, children? One is initiation. Next step is elongation. And the third step is termination. So, the 3 steps of transcription are initiation, elongation and termination. If you remember replication, there will tell in replication so many enzymes are required, right? So, for unwinding the DNA helix, DNA helicase enzyme is required. Then for starting the process initiation, DNA polymerase cannot do, then primase is required. Then for continuing the process, DNA polymerase is required. Isn't it? So many enzymes I am taking the list. But unlike replication, if you come to transcription, in transcription, one enzyme, that is RNA polymerase, is capable of doing all the roles. Okay, so let us write down that. Unlike replication where unwinding is done by DNA helicase enzyme. In replication, unwinding is done by DNA helicase enzyme. Then initiation of replication is done by primase enzyme, RNA primase. Then elongation of replication is done by DNA polymerase enzyme. So, in replication, each step is mediated by one enzyme. But in transcription, in the process transcription, we tell that one enzyme. What is that one enzyme? RNA polymerase. RNA RNA polymerase is capable of doing all the roles. Is capable of doing all steps or all roles. Children, in NEET 2020, there was a question on this concept. They asked, opening of the DNA helix during transcription is mediated by. So, what is the question? The question is, this is NEET 2020 question. The question is, opening of the DNA helix during the process of replication is mediated by. I can make the question as simple also, and they can ask you, unwinding of the DNA during replication is by. Unwinding of the DNA during replication is by. So that is a short sentence. Then we'll straightly think on it. But in the question paper, they have to twist you. They have to take the two keywords at this corner and at this corner. What they have done? They asked you, opening of the DNA helix during the process of replication is by. During the process of question is transcription is by. During the process of transcription is by. So, in hurry, we read the question fast. Then we think that opening of the DNA helix, I know it's DNA helicase. So, options also you know. So, RNA polymerase enzyme will not be in the first option. DNA helicase enzyme will be in the first option. Some other options are there. So, in replication, it's by DNA helicase. But in the question they asked you during transcription. Here I wrote, in transcription, one enzyme will facilitate all the steps. RNA polymerase only will open. RNA polymerase only will initiate. RNA polymerase only will elongate and terminate also, right? Then what is the answer? What is the answer? Who will open the DNA helix during transcription? This is the answer. But in hurry, if you don't understand the question properly, what will you tick? This you will tick and come. Gone. So be careful. Okay, so this was the question which has been asked in NEET 2020 examination. So where we are? We are with 6.5.3 prokaryotic transcription. So I told in this lecture we'll do prokaryotic transcription. In the next lecture we'll go to eukaryotic transcription. So it will give a clear idea for you. Prokaryotic transcription means bacteria. Then I told location. We draw a bacterial cell. There is no nucleus. There are no membrane-bound organelles. The DNA is naked, lying in the cytoplasm. So replication also here itself. DNA to RNA making also here itself. So location will be cytoplasm. The direction of replication. The RNA is synthesized from 5' to 3'. So polarity 5 to 3'. Enzyme required for transcription is called as DNA Dependent Rna Polymerase. One enzyme of bacteria will do the synthesis of all the 3 RNAs. What are the 3 RNAs? mRNA, rRNA, tRNA. mRNA will copy the message from the DNA. tRNA will carry the amino acids for the protein synthesis. rRNA makes the structure called ribosome where translation will happen, and it is catalytic in nature also. During translation, it acts as a catalyst also. Then, what is the name of the enzyme? DNA Dependent Rna Polymerase. Is it a monomer made up of one subunit, made up of many subunits? It is made up of many subunits, so it is a polymer. What are the subunits it is made of? 2 alpha, 1 beta, 1 beta prime, 1 omega. 2 alpha, 1 beta, 1 beta prime, 1 omega. 5 together is called core enzyme. This core enzyme called RNA polymerase, it shows transient temporary association either with the sigma factor or with the rho factor. Then what is it called? Holoenzyme complete enzyme. So the core enzyme with sigma factor helps in initiation. The core enzyme with the rho factor helps in termination. Coming to the steps of translation: initiation, elongation and termination. Now in replication, we told it's a complex process and one enzyme will come and do its function. So one enzyme for unwinding, one enzyme for initiating, another enzyme for elongating, like that. But in transcription, one enzyme has to tackle all the roles. It only has to unwind the DNA, it only has to initiate the process, it only has to elongate and terminate also. So in transcription, one enzyme RNA polymerase is capable of doing all the steps, and I gave a question which appeared in NEET 2020 on this concept, and then we saw it's not this, it is this. The answer will be this if they are asking you what is that enzyme which opens the DNA helix during replication. But in the question, they asked you what is that enzyme which opens the DNA during transcription. If it is transcription, this is the enzyme. If you want to take a screenshot, yeah, you can. Yes. Now we'll see the steps one by one in detail. Now, if you want to see the steps I told, the 3 steps are there: initiation, elongation, termination. The direction of transcription is downstream. There's a region called promoter which is upstream, right? So now we have to go with initiation. First step. Initiation is the first step in transcription. I told you RNA polymerase requires sigma factor to locate the promoter region. So the RNA polymerase enzyme, RNA polymerase binds with which factor? Binds with the sigma factor. This is sigma. It binds with the sigma factor to locate promoter region. To locate the promoter region specifically. RNA polymerase has to perfectly locate the promoter region. Okay. Now, where is the promoter? Promoter is upstream, right? It's at the 5' end of the coding strand. So, the promoter is upstream. Upstream. It is at the 5' end of the coding strand. Promoter is at the 5' end of the coding strand, right? Now, I told you prokaryotic promoters are separate, eukaryotic promoters are separate. So, what are the prokaryotic promoters? Since it is upstream, we have given the numbers as -1, -2, -10, -30, -35. There are 2 promoters in prokaryotic sequence. One is at -10 region, it's called -10 sequence. The other one is at -35 sequence. -10 sequence is a hexameric sequence. There are 2 promoters in bacteria. One promoter is at -10 sequence and the other promoter is at -35 sequence. -10 sequence promoter is called as Tata box, or it's called Pribno box. -10 sequence is called, we name it as Tata box, or we can also call it as Pribno box. It has a 6 base pair sequence. It has a 6 base pair sequence called Tata 80. It has a 6 base pair sequence called Tata 80. -35 region, they did not give any name for that, but then it also has a hexameric sequence called TTGACA. So we don't have any name, but it is also a 6 base pair sequence only. So it is TTGACA. The sequence is Tata 80, Tata box, Pribno box. The sequence is TTGACA, so which is at the 35 base pair sequence. Now, these promoter sequences are identified by the sigma factor. Sigma factor helps in locating these promoter sequences and it makes the RNA polymerase to come and bind to the DNA at that place. So, then the initiation will start. In the absence of the sigma factor, what happens? In absence of sigma factor, the specificity is lost. Then the RNA polymerase can come and bind on the DNA at any place, okay, randomly. So, we wrote here, like the RNA polymerase binds to the sigma factor and it locates the promoter region specifically if sigma factor is absent. If at all sigma factor is absent. So, let us write that condition also. If sigma factor is absent, then RNA polymerase binds on the DNA non-specifically and it starts from anywhere. So, if the sigma factor is absent, RNA polymerase binds to the DNA. Binds to DNA specifically na non-specifically na? Non-specifically. It binds to the DNA non-specifically at any place. At any place it will bind and it will do the process. So then sigma factor is very much required then. So here the sigma factor comes and binds, identifies the promoter region, it comes and occupies the DNA. Then when it occupies the DNA, then somehow we can tell that somehow the RNA polymerase, we'll write it in a better manner. After it comes and binds, locates and binds, somehow we know that RNA polymerase only has to do everything. Somehow RNA polymerase unwinds the DNA. It unwinds the DNA to read the sequence present on the template. To read the sequence, ATGC sequence is there? No. To read the sequence present on the template. Present on the template. So the sigma factor helps in binding. If we have to draw, we'll draw it. Come on. Yeah. If you have to see the sequence, minus 35 sequence is, I'll use this place, minus 35 sequence is T, T, G, A, C, A. Now we will see the process of transcription. In process of transcription, they have used two colors in our NCERT. Okay, for template stand they use this color. For coding stand they use this color. Template stand should be 3 to 5. So 3 to 5. Coding stand should be 5 to 3. 5 to 3. In the textbook, for the promoter region, they used yellow color. So we don't have yellow color. Let me go with black. They told this is the promoter. This is the promoter. And now just now we saw that RNA polymerase, its association with the sigma factor helps in locating the promoter. So RNA polymerase, core enzyme with the sigma factor helps in locating the promoter. It helps in locating the promoter. Now, this is a diagram. We have to show it for initiation. What is this step? If you have to write this step, you have to write it as initiation. You have to write it as initiation. They showed the hydrogen bonds also, children. This is how the DNA is. This is how the DNA is. And what is this step did I draw there? The sigma factor, locating the promoter, going and binding there. This step is called initiation. Step 1, initiation. Shall I write it here? Step 1 is initiation. Now, step 2 is elongation. Means in elongation, like you have to move from here to here. Means you have to unwind the next region. In transcription, not the complete DNA will unwind at a time, step by step, ring by ring, helix by helix, it will unwind. So now let us draw that also. What is that? Elongation. In elongation, we have to show right? Right. This is 5 to 3. Let me use one color itself. It's taking time. Right. Now, from promoter we crossed. From promoter we crossed. We moved to the next 2 turns, right? So then what is this step called? This step is called elongation. This step is called elongation. In elongation means DNA dependent RNA polymerase has opened it. You can see that it is opened up. it is opened up and it's doing the RNA synthesis. Step by step, it's doing the RNA synthesis. Now, at initiation only sigma factor is required. When you are moving away from the promoter region, that means during elongation, sigma factor is not required. That means here what happens, the rho sigma factor drops down. So, here if we see, the sigma factor drops down. The sigma factor drops down and only the remaining part, what we told as a core enzyme. Core enzyme will be continuing the elongation process, and this is the RNA which is getting synthesized. This is the RNA which is getting synthesized. A portion of the RNA is attached to the enzyme. See, a portion of the RNA is attached to the enzyme. And what enzyme is doing the elongation? Only core enzyme, only RNA polymerase. Sigma factor came down because you moved from this region, right? So, these are the bases which are exposed. Now, what is this? This is a RNA molecule, and this is our step for elongation. In termination, if we have to see. So, for termination, again we need to draw. Means this got transcribed, this got transcribed, this got transcribed, this also got transcribed, and oh, I need to use another color. So, blue color we are using, yes. Coding strand we are representing by blue, no? And for terminator, in NCERT they used green color. For terminator in NCERT we are having green, so we will use green. So, this is the terminator sequence. Here, this is the promoter sequence. This is a terminator sequence. Now, to identify the terminator sequence, we need rho factor. For promoter sequence, we need sigma factor. For terminator sequence, we need the rho factor, right? So, three prime, 5 prime, 3 prime. This is 5 prime. This is 3 prime. And for the blue, it is 5 prime. It is 5 prime. Now, terminator sequence has reached. Now, RNA polymerase will come here. Now, RNA polymerase should have a transient association with the other factor called rho factor. RNA polymerase, now it is having transient association with the rho factor. Now, this rho factor, what it will do? This rho factor stops the process. It pulls out the nascent RNA. RNA drops down. RNA drops down. RNA will drop down. Then afterwards, this enzyme also will come out. We can show directly the enzyme out itself. The rho factor comes and identifies this, and it drops down the enzyme also with the rho factor. Process stopped, process completed. So, this is initiation, this is elongation, then this step will be termination. This is the third step. What is the third step called? Termination. This step is called termination. So, RNA polymerase is doing elongation, and initiation, sigma factor will help in recognizing the promoter sequence. Rho factor will help in recognizing the terminator sequence, and this is the process which happens. So, during initiation we told RNA polymerase binds to the sigma factor. RNA polymerase binds to the sigma factor. Let me write down. So, what is this green colored one? This green colored one is RNA polymerase enzyme. It is RNA polymerase enzyme. RNA polymerase interacted with the sigma factor, became a holoenzyme. They can locate the promoter sequence. In the absence of this sigma factor, anywhere it binds and it starts the process unspecifically. Promoters in bacteria, there are two types, -10 sequence, -35 sequence. -10 sequence has a sequence of Tata AT, -35 has a sequence of TTGA-CA. Now, at this sequence, you need to open the helix. You need to open the helix. T and A has 2 bonds each. So, G and C has 3 bonds each. So, Tata AT, the bonds are less. So, you can easily open. So, it gets opened up, it unwinds. The sigma factor, if it is absent, non-specifically anywhere the process can start. Okay. So, then we have seen the picture. If you want to take the screenshot of this, take down, then we'll go to elongation and termination. Right? So, now if we have to write the steps of elongation and termination. In elongation, the enzyme will use nucleoside triphosphates as the substrates. The enzyme will use nucleoside triphosphates as the substrates to build the RNA molecule. Yeah, come on. Next step is elongation. During elongation, the sigma factor drops down. Just now I showed you in the picture, the sigma factor drops down and the core enzyme will do the elongation. And the core RNA polymerase continues elongation. The core RNA polymerase continues elongation. Okay. Sigma factor drops down and the core RNA polymerase continues elongation. The enzyme uses nucleoside triphosphates as substrates. NTPs, right? So, enzyme uses nucleoside triphosphates. What do you mean by nucleoside triphosphates? ATP, CTP, UTP, GTP as substrates. It uses these as substrates, as a building blocks. And it polymerizes the RNA, it synthesizes the RNA and it polymerizes the RNA molecule. Understood? Now, elongation works on the basis of base complementarity. A is there on the DNA, U will come. G is there on the DNA, C will come. So, elongation works on the principle of what? It works on the principle of base complementarity. Now, children, the third step, termination. The third step is termination. When the enzyme reaches the terminator sequence, we need Rho factor. When enzyme reaches downstream, right? When enzyme reaches terminator sequence, Rho factor identifies. Rho factor binds with RNA polymerase. Rho factor identifies the sequence. Now, you know, there are two types of terminations. Rho dependent termination, Rho independent termination. Sometimes if Rho factor is not there, then also termination will occur. What is such a type of termination? Called Rho independent termination. If the termination is mediated by the Rho factor, it's called Rho dependent termination. So, termination is of two types. What is that? Rho dependent termination is one type. The other one, in absence of Rho also it happens, that's called Rho independent termination. That's called Rho independent termination. Now, the Rho factor comes and what it will do? The Rho factor pulls the nascent RNA out. The Rho factor with RNA polymerase drops down during termination. Then RNA also drops down. Rho factor drops down, RNA also drops down. This is what is termination. This is what is termination. Understood? So, we have seen initiation, elongation and termination. Now, the RNA which is formed immediately after transcription. The RNA which is formed immediately after transcription, it's prokaryotic RNA. It is prokaryotic RNA. Children, the prokaryotic RNA is very much active. It is active. It is active and it doesn't have no split gene concept also. Split genes are not there. So, means no need to do any modifications. Since introns are not there, only exons are there, only coding sequences are there, no split genes are there, no nucleus and cytoplasm, no nucleus and cytoplasm separations. So, everything is happening in the common compartment called the cytoplasm itself. In eukaryote, transcription inside the nucleus, translation in the cytoplasm. But in bacteria, everything is in the same compartment, right? So, no nucleus and no cytoplasm separations in bacteria. And again, no processings also, because everything is happening there. No capping, no tailing, no splicing, no introns. So, no introns, no nucleus and cytoplasm separation, and the RNA is active. So, before the transcription ends, translation will start. So, before transcription ends, what will start? Translation starts. Before transcription ends, translation starts. Means transcription did not still reach the terminator. Transcription did not still reach the terminator. Translation means it happens on the ribosomes. So ribosomes meanwhile come and they'll tag here. Ribosomes, they'll come and they'll tag here. They want this region. They can synthesize the protein like this. They can synthesize the protein like this before the transcription got completed. So we tell that, so in eukaryotes, transcription, translation are coupled. Means before the transcription ends, protein synthesis, translation starts. All are favorable to start translation because everything happens in the same compartment, so it can occur. There are no split genes, so there is no processing required, it can occur. And the RNA which is formed immediately after transcription, which we call primary transcript, the mRNA, is very much active. It need not need any processing. So that is why we tell in bacteria, transcription and translation, means protein synthesis, are coupled. Transcription and translation are coupled. It is not possible in eukaryotes. It's only possible in prokaryotes because of same place everything is occurring. So with this, we have completely done prokaryotic transcription. If we have to revise, so in prokaryotes, one enzyme will do the synthesis of all the three: initiation, elongation, termination. It can unwind also, it can start also, it can elongate also, it can terminate also. Its temporary association with the sigma factor helps in identifying the promoter starting the process. Its temporary association with the rho factor helps in identifying the terminator sequence and stopping the process, right? So the promoter regions are of two types: -10 base pairs and -35 base pairs sequence. -10 base pairs is TATA AT sequence. -35 base pair sequence is TTGS CS sequence. Sigma factor will identify. Elongation, you need the building blocks, NTPs, ATP, CTP, UTP, GTP will come and they will elongate. When the termination region reaches, rho factor will come, bind to the RNA polymerase, stops the process, pulls everything out. And the RNA which is formed after that is active. There are no split genes. Everything happened in the same place. So then before the transcription ends, translation will start. So this we tell like transcription and translation are coupled in bacteria. Hope you understood the lecture. Is it interesting? So any queries you can post me in the comment box. So in the next session, we will talk about eukaryotic transcription. Thank you.